This video, we will cover basic knowledge in cell culturing, including important equipment, how to use a biosafety hood, and proper aseptic technique. You will need a lab coat, a pair of goggles, and disposable gloves. They can be nitrile or latex. Nitrile is often preferred because they are more resistant to chemicals. 70% ethanol is the main disinfectant you will use for mammalian cell culturing. Always keep a bottle near you. Lastly, you have the biosafety hood. The hood is kept sterile by laminar flow traveling top down to the front grill and to the back vent. The following test demonstrates the pathway of air inside the biosafety hood. Notice airflow splits at the surface before traveling straight across into the back vent and the front grill. Beware that air from outside also gets into the biosafety hood. They get sucked into the grill, but it is still best to work with little movement outside near the biosafety hood.
In this section, we will learn how to prepare the biosafety cabinet for use. First, wear gloves and clean with 70% ethanol before entering the hood every time. When opening the hood, be sure to open it to the correct height as shown on the side of the hood. Before using the hood, be sure to let it sit for 5 minutes to allow the air to circulate. When using the biosafety hood, make sure to turn the light on and wait for the performance meter as shown by the two lights at the bottom to turn green. Next, make sure to spray down the bottom of the hood with 70% ethanol and wipe with a Kim wipe. When bringing things into the hood, be sure to spray down with 70% ethanol and wipe thoroughly. Do this with all items brought into the hood. This will help reduce any sources of contamination that could possibly be brought into the hood on these items. Be sure to keep the vent at the back of the hood and the front clear of items, because it could block the airflow, keeping both the cells and you protected. Many times, things need to be warmed up before being used in cell culture. This is done by placing them in the 37 degrees Celsius water bath. Be sure to dry them thoroughly and clean with ethanol before placing in the cell culture hood. Bring only items needed for a particular procedure in order to prevent cluttering your workspace. This is an example of what not to do. Now we will go over how to work in the biosafety cabinet. Be sure to designate areas for each type of material. Working materials like cells in the front, waste container and tools such as pipettes on the sides, bottles, containers, and caps in the back. When opening anything in the hood, open it from the side. The inside of the container is considered sterile. Any movement over these open items could contaminate them. Lids should be placed toward the back of the hood, face up, in the same order that the containers are placed in the rack. When opening a well plate, grip with 2 fingers and open from the side. Do the same to close it. Never pour from one container to the next. This could introduce contaminants into the liquid. Instead, use an auto pipetter. To use an auto pipette, open the pipette tip with the bottom half outside of the hood. Place the tip of the pipetter onto the auto pipette and then orient so that you can easily read the volume indicators. Handle the pipette with a steady hand. Avoid large motions and do not let the tip touch anything non-sterile. This includes the bottles. Always use at an angle to avoid contamination. Try to avoid any bubbles, as these can lead to more contamination. Do this by pouring onto the side of the bottle. Once done with the pipette tip, dispose of it by putting it back into its wrapper and then placing onto the side bin. Avoid a collection of trash. When using a micropipetter, open the container slowly and select a pipette tip from the side while avoiding going over any of the others. When aspirating liquid, use the aspirator tips from an autoclaved metal box. Treat the box like a sterile container and remove the tips from an angle. Place the end of the tip onto the aspirator tube, like so. Do not allow the tip to touch anything inside of the hood. Close the box at an angle. Be careful of the glass aspirator tips inside. The pump is located beneath the hood. Turn it on like so. When removing things from the hood, be sure to remove the cells first, followed by any chemicals or cell media. Next comes the tools. Last are the bottles and the waste. To remove the glass aspirator tip, slowly twist the glass inside the tube until it removes. Then spray down the tube with ethanol. To get rid of the aspirator tip, place it into the sharp biohazardous waste next to the hood. After everything is removed from the hood, be sure to spray it down and wipe it clean with ethanol. Any waste that touched cell media or cells or chemicals need to be placed into the autoclave black and red bags. After you are finished with the hood, be sure to close it to the correct height, again labeled on the side. Remember to turn off the light in the hood and turn on the UV light. The UV light is used to sterilize the hood. We put it on for 30 minutes after use. Do not rely solely on the UV light because the bulb could be faulty.